Now, one of the things I'm going to talk about before we get into talking about the mechanisms of gene transfer is something called homologous recombination. Now, when we're talking about moving genes from one bacteria to another, in order to have them expressed in the recipient bacterial cell, you have to almost always, and there are exceptions that we'll go over, but almost always you need to have this process of homologous recombination. Okay? So we get a piece of DNA, and in order for it to be expressed, it's going to have to exchange with a homologous area of the genome. All right? So this is a really important process. So let's look at the process, homologous recombination. We've got, I love to think about gene transfer as kind of shopping for genotypic and phenotypic properties that you like, right? Let's say you want a nose job. Let's say you want a reduction, or let's say you want something else. You could go shopping for genes that would help you do that. So you pick the gene that you want, and it's going to come in to the cell, find an area that it's homologous to, and it's going to exchange or recombine with the DNA that's already there. So that's part of the rule here for homologous recombination. It has to find an area of homology with DNA on the strand of the bacteria. If that happens, we can then undergo the recombination event using several proteins, but with Rec-A being the absolute requirement for homologous recombination, we can allow exchange for that genetic information. So let's look at the example here of homologous recombination. If you're looking at the cell here, you can see the white circle is analogous and supposed to represent the bacterial chromosome. The blue strand of DNA that you see coming in here is a linear piece of DNA that has been taken up by this bacteria. Now, what has to happen is we have to find region of homology between this blue strand and the chromosomal DNA. So what happens is we have coming in on that strand of DNA, we've got an A- allele and a B- allele. So it's to find something that it's similar to. So it goes to the alleles that it's most like, and that would be the A+ and the B+ alleles that you see here. And with the help of Rec-A, we'll then exchange DNA. So if you look in this other cell, after recombination, what you have is you have now the alleles that were on that linear piece of DNA in the chromosome, and you will have lost the alleles that were exchanged. So you're not adding a piece of DNA, you're really exchanging DNA. So this DNA is not going to become larger from this homologous recombination. We're just exchanging alleles. And the alleles that we have exchanged for are no longer there. We're no longer going to have access to them. That is quite different than site-specific recombination. Site-specific recombination is an integration of DNA. So if you're looking at site-specific recombination, for example, you have a large red circle here, the blue circle here, and for example, the large red circle integrates with the blue circular DNA. Now we have the addition of those two pieces of DNA. So the molecule now is a sum of the red circle and the blue circle. What are the three major roles here of site-specific recombination? Integration of a fertility factor, which we'll talk about in a little bit. Integration of bacteriophage into a bacterial chromosome, and movement and insertion of transposons into DNA. Remember, this is going to be an addition, so the molecule becomes a sum. It actually gets bigger. That's very different than homologous recombination, where we're simply exchanging those strands.